Between 2014 and 2016, an infectious outbreak in West Africa claimed over 11,000 lives, devastating the healthcare infrastructures of Guinea, Liberia, and Sierra Leone. This photograph captures the societal strain of the epidemic, reducing the fight against the Zaire Ebola virus species to a grim public scoreboard. The pathogen dismantled both the biological systems of its hosts and the social order of entire communities. Ebola virus disease, or EVD, is a severe zoonotic illness. Clinically, it is defined by initial nonspecific symptoms that rapidly escalate into gastrointestinal distress and multi-organ dysfunction syndrome. The virus originates via zoonotic spillover. Its natural reservoir exists within fruit bats of the Pteropodidae family. When humans encounter the blood or bodily fluids of infected animals in the rainforest, the pathogen crosses the species barrier. Once inside a human host, human-to-human transmission occurs via direct contact with infected fluids, leading to a rapid clinical decline that ultimately culminates in severe distributive shock. The agent responsible for this systemic collapse belongs to the orthebovirus genus. This electron micrograph isolates the physical pathogen. The enveloped single-stranded RNA virus is distinctly filamentous, often twisting into characteristic U or 6 shapes, measuring about 80 nanometers in diameter and extending up to 14,000 nanometers in length. A pathogen of this microscopic scale, entirely devoid of cellular machinery, triggers one of the most comprehensive systemic failures observed in human medicine. Genetically, the Ebola virus is extremely economical. It is a non-segmented, negative-sense, single-stranded RNA virus. This schematic maps the viral genome. At 19 kilobases, it is a fraction of the size of genetic sequences in complex organisms. From this short sequence, the virus codes for 7 structural and non-structural proteins, linearly ordered from nucleoprotein to RNA-dependent RNA polymerase. The human immune system deploys billions of highly specialized cells, intricate signaling cascades, and massive protein networks to defend the body. The virus defeats this architecture using only 7 distinct genetic tools. It achieves this through a highly coordinated 2-stage pathogenesis. First, the virus executes a stealth operation, deploying specific proteins to entirely blind and paralyze the host's innate immune response. Second, once replication peaks under the radar, the virus weaponizes the host's own cellular machinery, deliberately triggering a massive cytokine storm to dismantle the body's vascular integrity. The lethality of EVD does not rely on brute force. It relies on the precise, targeted sabotage of fundamental host mechanisms using a payload of only 7 proteins. The invasion begins with the virus selecting its primary cellular targets, the body's frontline defenders, specifically monocytes, macrophages, and dendritic cells. The viral glycoprotein binds to specific host surface lectins, such as DC sign, and phosphatidylserine receptors on the outside of the immune cell. Instead of injecting its genetic material immediately, the virus allows the host cell to swallow it whole through a process called macropinocytosis. The virus is now trapped inside a host endosome, surrounded by acidic fluid and digestive enzymes intended to destroy cellular debris. This animated cross-section of an endosome shows how the virus turns the host's defenses against itself. Host endosomal cathepsins, specifically cathepsin B and L, inadvertently assist the pathogen by cleaving the viral glycoprotein. This cleavage strips away the outer layer, exposing a hidden binding domain on the virus. This newly exposed domain immediately locates and locks onto the Niemann-Pick C1 receptor, located on the inner wall of the endosome. Securing the NPC1 receptor is the strict mandatory trigger for entry. Once bound, the virus fuses with the membrane and ejects its RNA directly into the host cytoplasm. The pathogen cannot initiate replication until it exploits host digestive enzymes to facilitate the conformational changes required to engage the NPC1 receptor and reach the cytoplasm. Under normal conditions, a cell detecting foreign RNA immediately produces type 1 interferon, a chemical alarm signaling neighboring cells to raise their antiviral defenses. This split-screen cellular model illustrates how the virus suppresses that alarm. On the left, the viral protein VP35 binds to the viral double-stranded RNA, physically masking it from the cell's internal center molecules. By hiding the viral RNA from recognition, VP35 prevents the activation of downstream pathways, completely halting the production of type 1 interferon. The internal alarm is disconnected. Simultaneously, the virus must defend against external alarms triggered by neighboring cells that may have detected the infection. Returning to the right side of our model, the viral protein VP24 binds to karyoferrin alpha at the host cell's nucleus, creating a physical blockade at the nuclear pore.
This blockade prevents phosphorylated STAT1, the messenger molecule for the interferon response, from entering the nucleus, rendering the cell entirely unresponsive to any circulating interferon in the environment. With the primary alarms disabled, the virus causes massive functional paralysis in dendritic cells. These infected cells fail to mature and cannot upregulate the CD80 and CD86 co-stimulatory molecules required for communication. Without mature, functioning dendritic cells to present antigens, the body experiences a complete failure to initiate an adaptive T-cell and B-cell response. As this simulation demonstrates, bystander lymphocytes are decimated. Although T and B cells are not productively infected by the virus, they undergo massive apoptosis, driven by Fas and trail signaling pathways, leading to profound lymphopenia. By deploying VP35 and VP24, the virus turns the body's primary alarm system into a silent, self-destructing void, securing time to replicate undetected. Once viral replication peaks, the stealth phase ends. Heavily infected macrophages transition from silent incubators into engines of systemic destruction. These dying macrophages release massive quantities of pro-inflammatory cytokines and chemokines into the bloodstream, specifically TNF-alpha, interleukin-6, and nitric oxide. This animated diagram of a blood vessel wall reveals the primary consequence of this cytokine storm: endothelial hyperpermeability. The chemical surge causes the tight junctions binding the vascular endothelium to physically break down. The resultant fluid shift forces intravascular volume out of the bloodstream and into the surrounding tissues, causing severe hypovolemia, third spacing, and ultimately distributive shock. Parallel to this vascular leak, infected monocytes express exceptionally high levels of surface tissue factor. As simulated here, this triggers disseminated intravascular coagulation, or DIC. Widespread microvascular thrombosis occurs throughout the body, rapidly depleting clotting factors and circulating platelets. The virus replicates heavily in hepatocytes, causing extensive reticulated hepatic necrosis. As the liver fails, the synthesis of new clotting factors halts entirely, severely exacerbating the consumptive coagulopathy. The profound lethality of Ebola virus disease is driven by the deliberate weaponization of the host's own immune and coagulation systems, resulting in an irreversible vascular collapse. Following an incubation period of 2 to 21 days, patients enter the initial dry phase, lasting roughly 3 days. During this phase, the virus is still executing its stealth replication. Clinically, this manifests as abrupt, highly nonspecific constitutional symptoms: high fevers, severe chills, myalgias, and profound asthenia. As the stealth blockade breaks down and viral load climbs exponentially, patients transition into the wet phase between days 4 and 9. This clinical timeline chart maps the intersecting biological forces. As the viral load rises, the cytokine storm triggers severe gastrointestinal manifestations. Profuse, watery diarrhea and projectile vomiting cause massive fluid shifts, leading to the plummeting intravascular volume you see here, often exceeding a loss of 5 to 10 liters per day. Patients surviving into the severe systemic phase at day 10 face the catastrophic results of DIC and volume loss. Clinical markers include anuria, encephalopathy, and profound metabolic acidosis, leading to coma and shock. The macroscopic, triphasic clinical presentation of the disease maps exactly to the escalating timeline of microscopic internal sabotage. Recognizing the virus early remains a distinct diagnostic challenge. The initial constitutional symptoms of the dry phase mimic many endemic tropical diseases. This blood smear reveals the purple parasites characteristic of severe malaria.
Clinicians differentiate EVD from severe malaria by noting Ebola's distinct lack of early splenomegaly and the absence of cyclical fevers, later verifying the diagnosis through rapid testing. EVD must also be separated from typhoid fever. Ebola patients do not exhibit the Faguet sign, a notable bradycardia relative to their fever, and lack the characteristic rose spots associated with Salmonella typhi. As the wet phase progresses, specific red flag signs predict immediate mortality. Tachypnea and deep Kussmaul breathing indicate profound decompensated metabolic acidosis, while anuria points directly to acute kidney injury from prolonged hypovolemia. While hemorrhagic manifestations like petechiae or mucosal oozing occur in roughly half of all cases, frank exsanguination is remarkably rare. Mortality is driven by decompensated shock and anuria, not by blood loss. Grasping the molecular pathophysiology of the virus allows clinicians to pierce its initial flu-like disguise. Anticipating the inevitable vascular collapse is the only way to initiate aggressive, life-saving fluid resuscitation before the onset of irreversible shock.